In this presentation, we'll look at how genes located on the same chromosome assort during meiosis and how researchers mapped their locations onto chromosomes using linkage mapping. In previous presentations, we've seen in detail how genes on separate chromosomes assort independently of one another. That is, the two genes do not influence each other in terms of which allele ends up in which gamete. As a result of this, all four of these possible combinations of alleles are equally likely. In particular, notice that both red chromosomes may appear in a gamete together, or they may appear in a gamete with a blue chromosome. Now, both red chromosomes were inherited by the F1 generation from the mother. That is, they're maternal chromosomes. The blue chromosomes were inherited by the F1s from the father and are therefore paternal chromosomes. We therefore call the gametes with both maternal or both paternal chromosomes parental types, and the gametes with a mixture of maternal and paternal chromosomes we call recombinant. As you can see, we expect half of the gametes to be parental types and half to be recombinant. But what happens when the two genes we're interested in are on the same chromosome? Well, if meiosis simply happens like this, then we would expect that both alleles that are on the maternal chromosome would be inherited together, and both alleles that are on the paternal chromosome would be inherited together. Based on this, we would expect to have all parental types and no recombinants. So, does this mean that if genes are on the same chromosome, they will always be inherited together as parental types? The answer to that is no. Just because they're on the same chromosome doesn't mean they'll be inherited together. The reason for this is that crossing over may separate them. Let's look at this example here with 3 genes on the long arm of this chromosome. The paternal alleles are all big letters, big A, big B, and big C. The maternal chromosome carries the 3 little lettered alleles. For now, let's just consider the A and C genes. So, any gamete that's produced will have one allele of the A gene and one allele of the C gene. If it gets both genes in the same combination as they were in one of the parents, then it has the parental type. And if it gets a new combination due to crossing over between the genes, then it has a recombinant type. You can see that in order to get recombinant types, there needs to be crossing over between those two genes. The A and C genes are pretty far apart, so crossing over between them is actually fairly likely. And so all of these gametes are produced in approximately equal numbers. Now, let's look at this again. And instead of focusing on the A and C genes, let's look at the A and B genes. Notice that these are closer together along the chromosome. In this particular meiosis, even though there's crossing over, the crossing over does not occur between the A and B genes, and so there are no recombinant gametes produced. All of the gametes are the parental type. In some cells, the point of crossovers may be between these two genes, so in those cases there would be recombinant gametes produced. However, on the whole, recombinant gametes would be much less common than parental types. If we take this to the extreme and imagine that the A and B genes were right next to each other, it would take a very rare event of crossing over in just the right spot to produce recombinant gametes. So almost all of the gametes would be parental. So let's summarize this. If the loci are distant, then crossing over, perhaps even more than once, is likely between the genes, and we expect 50% of the gametes to be parental and 50% recombinant. We should never expect more than 50% to be recombinant, as that's what it would be if the genes were on different chromosomes and were assorting completely independently. If the genes were closer together, then we'd expect to get somewhat less than 50% recombinant genes, as we'd need crossing over to be between the genes, and this would be somewhat unlikely. If the genes are very close together, then we might expect very few recombinant gametes, with most of them being parental. So if you understand this logic, you can see how researchers created genetic maps to map out the location of the genes relative to one another. How do we actually count how many gametes are recombinant and how many are parental? We can't just look at lots of eggs and sperm and identify them as parental or recombinant. Well, the easiest solution is to do a test cross using a tester that is homozygous recessive for both traits. We can then easily identify the offspring that are from the parental type gametes and those that are from the recombinant gametes because we know that recessive alleles must have originated from the tester. Let's imagine that we do this experiment and we count a total of 1,000 offspring. We identify 900 of them as being parental types and 100 as being recombinant. We therefore say that the distance between these two genes is 10 map units. The number of map units is simply the percentage of recombinant types produced. We can use map distances in order to identify the order of genes. In this example, we find that the distance between genes A and B is 10 map units, 15 map units between A and C, and 5 map units between B and C. With this data, we can try to figure out the order of genes along the chromosome. If we try putting gene A between the other two, then the distances just don't work out. The largest distance that we have estimated is 15 map units, so we should put those on the map as far apart as possible. If we do that, the numbers add up properly. One little detail to say about this is that the map units often don't quite add up. Some of this may be due to random chance in the experiment, but there's also a chance of 2 crossovers happening between A and C. When this happens, it would make more of the offspring look like parental types, and so reducing the distance that you'd estimate between A and D to maybe, say, 14 map units. It's also possible between the closer pair of genes, but much less likely. Let's look quickly at another example. Here, a homozygous wild-type fly was mated with a homozygous doubly recessive fly to produce an F1 that is a dihybrid. In this case, we know that the 2 wild-type alleles are on the same molecule, and the 2 recessive mutant alleles are on the same chromosome. Those dihybrids are then crossed with another set of mutant homozygotes. The dihybrid F1s undergo meiosis to produce gametes. During meiosis, there may be crossing over between these 2 genes. If there is crossing over, recombinant gametes will be produced. When there's no crossing over, parental gametes will be produced. Notice that there's no crossing over in the tester because it's a homozygote at both loci. So even if there was crossing over, you wouldn't know about it. As a technical note, there's actually never any crossing over in male Drosophila, a fact that can make carrying out experiments like this easier. However, crossing over is generally the rule in diploids, so this is an oddity of Drosophila. Here, you can see the different types of gametes produced and how, because the mating is with a homozygous recessive, it produces phenotypes that can easily be assigned to genotypes. This allows us to identify how many meiotic divisions involved crossing over between the 2 genes. The gray normal winged flies and the black vestigial winged flies are the parental types. These other 2 phenotypes are recombinant because crossing over has occurred between them. Finally, we simply add up the number of recombinant types and divide by the total number of offspring that we've counted to get the recombination frequency. This is then the same as the number of map units. In this case, we get 17% recombination and 17 map units.